Every day, in order to interact with our environments, we have to move. And the way our brains generate this function is through kind of electrical and chemical communication. Today, I'm going to really narrowly talk about brain waves in the so-called beta band, which are frequencies from 13 to 30 hertz over the sensory motor cortex throughout movement. And by 13 and 30 hertz, I would conceptualize this as if you have white light passing through a prism and you'd have the different colors through the other side. You could, by studying the different bands of colors that come out, would be analogous to electrical signal and then studying the different frequency bands. So throughout movement, we can see beta power, which would be the amount or saturation of each one of these colors in our analogy, that before movement, we see that there's beta decrease, which is maximally decreased at movement, and then a beta rebound after movement. However, Parkinson's disease patients have characteristically slowed movement and elevated beta band activity in between the thalamus, the cortical areas, and the basic ganglia. And so my project looks at how is beta power modulated by movement speed in healthy participants to see if we can see some analogy between Parkinson's disease patients. And our hypothesis was that the magnitude of beta power, that being how low it gets and then how high it rebounds, is reduced on our so-called slow blocks. So we generated slow blocks in order to operationalize movement speed through a simple task. And so there's a black circle and then an arrow which indicate to get ready and then go move. And a short break and repeat. We can see that with a button press. However, if we had this response period be longer, that this would be our slow block and shorter would be our fast block. The idea being if you have more time to respond, you will respond more slowly. And similarly, as kind of this additional question, which we won't get too much into today, if this 4 period has a consistent length, you'll be able to predict when to move versus in this changing condition. And so looking at the reaction times, see how quickly people responded, we saw in the slow blocks that there were longer reaction times than in the fast blocks. Okay, fine, the task works. The next step here is to record with electrophysiology, in this case, electroencephalography, which is electrodes placed on the scalp to record the electrical brain signal discussed in the introduction. And so what we found was, so we have the fixed and fast for, the slow and fast for the fixed and varied conditions. And by looking between them, we can see just how movement speed manipulates these brain waves. And while this is a little more complicated than the simple trace we had before, we can see that this decrease indicated by the blue, seeing the difference between the slow and fast blocks, and again, this blue between the slow and fast blocks, that's the reduced rebound, except now that we can actually see all of the frequencies between 13 and 30 versus the kind of averaged version seen in the introduction. And so the easier way to kind of look at this is by averaging between 13 and 30 hertz to get you just a mean value. And we can really see, while perhaps in the context of the fixed blocks, that being the consistent 4 periods, that there aren't significantly differences between the two, we see in the slow versus fast blocks that this reduced rebound is conserved, that being this red trace being lower than this blue trace. So while this is interesting on its own, we actually can see from a previous study done in 2014 out of University of Nebraska, looking at beta activity between control groups and Parkinson's disease groups explicitly. And the big takeaway that's similar to our results is we can see the beta rebound for controls is actually greater than that than the beta rebound seen in Parkinson's disease, which is quite analogous to our findings. And this suggests that in our study, we actually had subjects in a so-called slowed movement state, as we call it, maybe due to these relaxed task constraints, but it shows that whether in normal people or in folks with Parkinson's disease, we can see some analogy between the slowed movement.